Let's begin with the most fundamental unit from which all imaging findings ultimately arise, the cell. Cell is the basic structural and functional unit of life. The human body contains approximately 100 trillion cells. Each cell performs essential functions including metabolism, energy production, and genetic regulation. The living content of the cell is called protoplasm. It is composed mainly of water, proteins, lipids, and carbohydrates. Water forms the major component and acts as a medium for biochemical reactions. Proteins function as enzymes and structural elements. Lipids form the cell membrane and participate in signaling. Carbohydrates serve as the primary source of energy. Structurally, the cell is divided into three main components. The cell membrane acts as a selectively permeable barrier, regulating transport. The cytoplasm contains organelles and is the site of metabolic activity. The nucleus contains DNA and controls cell division and protein synthesis. Let us now focus on the cell membrane, a dynamic interface between the cell and its environment. The cell membrane is primarily composed of a lipid bilayer, which provides structural integrity and fluidity. Embedded within this bilayer are integral proteins that span the membrane, while peripheral proteins remain attached to its surface. Carbohydrates project outward and are essential for cellular recognition and signaling. Functionally, the membrane regulates the movement of substances across it. Selective permeability allows controlled exchange of molecules. Active transport mechanisms utilize ATP to move substances against concentration gradients. Membrane proteins act as channels and receptors, facilitating communication between cells. The membrane also anchors the cell to its surrounding matrix. Integrins connect the cell to extracellular components such as fibronectin and collagen within the basement membrane. Growth factor receptors present on the surface bind external signals and regulate cellular responses. From a radiology perspective, all imaging findings originate at the cellular level. Disease begins with alteration in normal cellular structure or function. In tumors, there is uncontrolled cellular proliferation. In metabolic imaging, such as FDG PET, increased glucose uptake reflects increased cellular activity. Membrane components act as crucial diagnostic targets. Tumor characterization depends on membrane receptors and surface markers. Estrogen receptors in breast carcinoma and CD markers, such as CD117 in gastrointestinal stromal tumors, are key examples. Thus, understanding the cell and its membrane forms the foundation for interpreting every normal and abnormal imaging pattern in radiology.